you.
NCERT Class 11th Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done extensively, not using the criteria that were scientific, but born out of the need of use of organism for our use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom were developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants, animals were easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, mode of nutrition, nature of wall and habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Though the plant and animal kingdom have been a constant under all different systems, the understanding of what group the organism be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whitaker proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system also have been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic organism in the third domain and thereby a six-kingdom classification. You will learn about the system in detail in higher classes. Let us look at the five-kingdom classification to understand these issues and considerations that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, and the angiosperm under plants. The criteria that unified this whole kingdom was that all the organisms included has a cell wall in their cell. This placed together groups which were widely different in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones, say for example Chlamydomonas and Spirogyra were placed together under algae. The classification did not distinguish between the heterotrophic group fungi and the autotrophic green plant, though they also saw a characteristic difference in their cell wall composition. The fungi had chitin in their cell wall while the green plant had cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella earlier placed in algae within Plantae and both have cell wall, with Paramecium and Amoeba which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in early classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of change will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt have been made to evolve a classification system which reflect not only the morphology, physiology and reproductive similarities but is also phylogenetic, that is based on evolutionary relationship. In this chapter we will study characteristics of kingdom Monera, Protista and fungi of the Whitaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow and deep ocean where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, plural cocci; the rod-shaped bacillus, plural bacilli; the comma-shaped vibrio, plural vibrio; and the spiral spirulum, plural spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from the inorganic nutrients. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs, thermoacidophils and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffalo, and they are also responsible for production of methane, that is biogas, from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, that is nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrite and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legumes, roots, etc. Some are pathogens causing damage to human beings, crop, farm animals and pets. Chlorella, typhoid, tetanus and citrus canker are well-known diseases caused by different bacteria. Some bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by some sort of sexual reproduction by adopting primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with Plantae, Animalia, and fungi. Being eukaryotic, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelle. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving Cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae desmids. They are found in freshwater as well as marine environment. They are microscopic and float passively in water current planktons. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping cells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus diatoms left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used for polishing, filtration of oil and syrup. Diatoms are the chief producer in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cell. The cell wall has stiff cellulose plate on the outer surface. Most of them have two flagella, one lie longitudinal and the other transversely in a furrow between the cell plates. Very often red dinoflagellates, example Ganyulex, undergo such rapid multiplication that they make the sea appear red tides. Toxins released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which make their body flexible. They have two flagella, one short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other organisms, smaller organisms. Interestingly, the pigment of euglenoids are identical to those present in higher plant, example Euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body move along decaying twig and live engulfing organic matter. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiate and form fruiting bodies bearing spores at their tip. The spores possess true walls. They are extremely resistant and survive for many years under adverse conditions. Spores are dispersed by air current. 2.2.5 Protozoans. All protozoans are heterotrophs and live as producer or parasite. They are believed to be primitive relative of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater or moist soil. They move and capture their prey by putting out pseudopodia, that is false foot, as in amoeba. Marine form have silica cells on their surface. Some of them, such as int amoeba, are parasites. Flagellated protozoans. The members of this group are either free living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness, example trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity called gullet that open to the outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with food to be stirred into the gullet, example paramecium. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium malariae, malarial parasite, which cause malaria, a disease which has a stunning effect on human population. 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They saw a great diversity in morphology and habitat. You must have seen fungi on a moist bread or rotten fruit. The common mushroom you eat and toads are also fungi. White spots seen on mustard leaves are due to the parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Poxenia is also an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, and soil and on animals and plants. They prefer to grow in warm and humid places. You have even wondered why we keep food in the refrigerator. Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which is a unicellular, fungi are filamentous. Their body consists of long cylinder thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cynocytic hyphae. Others have septa or cross walls in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substances and hence are called saprophytes. Those that depend on living plant and animal are called parasites. They can also live as symbionts in association with algae as lichen and with root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and sexual reproduction is by oospore, ascospore and basidiospore. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps: fusion of protoplasm between two motile and non-motile gametes called plasmogamy, fusion of two nuclei called karyogamy, meiosis in zygote resulting in haploid spores. When a fungi reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2n. However, in other fungi, Asco and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon and the phase is called dikaryophase of a fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occur, leading to the formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood in moist and damp places or as obligate parasite on plant. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospore motile or by aplanospore non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or oogamous. Some common examples are Mucor, Rhizopus, the bread mold mentioned earlier, and Albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes. Commonly known as sac fungi or Ascomycetes are mostly multicellular, example Penicillium, or rarely unicellular, example Yeast Saccharomyces. They are saprophytic, decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiospore. Conidia on germination produce mycelium. Sexual spores are called ascospore, which are produced endogenously in a sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarp. Some examples are Aspergillus, claviceps, Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and considered delicates. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bract fungi or puffball. They grow in soil, on logs and tree stump, and in living plant bodies as parasite, example rust and smout. The mycelium is branched and septate. The sexual spores are generally not found, but vegetative reproduction by fragmentation is common. Sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus mushroom, stelago sprout, and coccinia, that is rust fungi. 2.3.4 Deuteromycetes. The commonly known as imperfect fungi because they are asexual or vegetative phase of these fungi are known. When the sexual form of these fungi are discovered, they were moved into classes they rightly belong to. It is also possible that the sexual or the vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another, and the placed under other classes. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral recycling. Some examples are Alternaria, Polytrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are particularly heterotrophic, such as the insectivorous plant or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Life cycle of plants has two distinct phases: the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phase and whether these phases are free living or dependent on others vary among different group of plants. This phenomenon is called alteration of generation. You will study further details of this in chapter 3. Kingdom Animalia. 2.5 This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a defined shape and size. Higher forms of elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. 2.6 Viruses, viroids, prions, and lichens. In the 5 kingdom classification of Whitaker, there is no mentioning of lichen and some acellular organisms like virus, viroids, and prions. These are briefly introduced here. All of us have suffered the ill effect of common cold or flu. Know what effect virus can have on us, even if we do not associate it with our condition. Viruses did not find a place in the classification since they are not concerned truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell and replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Pasteur. D.J. Ivanovsky recognized certain microbes as causative organisms of mosaic disease of tobacco. These were found to be smaller than bacteria because they pass through the bacterial proof filter. M.W. Bezerinki demonstrated that the extract of the infected plant of tobacco could cause infection to healthy plant and called this fluid as Contagium vivum fluidum. Infectious living fluid. W.M. Stanley showed that viruses could be crystallized, and crystals consist of largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No viruses contain both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophage viruses that infect the bacteria are usually double-stranded DNA viruses. The protein coat called capsid, made of a small subunit called capsomere, protect the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric form. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in human is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Dinner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most noble disease caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant Crr-Jakob disease (CJD) in humans. Lichen. Lichen are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and the fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food from fungi and fungi provide shelter and absorb mineral nutrients and water from its parent for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichen are very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime mold, and protozoans. Protists have defined nucleus and other membrane-bound organelle. They produce both asexually and sexually. Members of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes are the four classes under this kingdom. The Plantae include all eukaryotic chlorophyll-containing organisms. Algae, bryophyte, pteridophyte, gymnosperm and angiosperm are included in this group. The life cycle of plants exhibit alternation of generation, that is gametophyte and sporophytic generation. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroids as well as lichen are not included in the five kingdom system of classification. Thank you.